It is a great honor to be here, and thank you all for the opportunity to make a brief opening remark about this Japan session's intentions. Uh, First of all, FRIM stands for the Forum for Innovative Regenerative Medicine. It has been established in 2011. Our mission is to accelerate industrialization of regenerative medicine. We have more than 250 member companies now. It consists of pharmaceutical companies, chemical and material companies, machinery device companies, logistics, insurance, and consulting companies. It consists of the very diversified business field of members. Firms have a global partnership by MOU in the field of regenerative medicine industry, including ARM. To maximize the opportunity internationally, we introduced Japanese companies at overseas event and introduce overseas companies at Japanese event. We think it is a priority issue to promote harmonization and optimization of the regulation of regenerative medicine product among Asian countries and the region. So we organize APAC-RM is to sort out point of difficulties toward harmonization now. The member of APAC-RM are these organizations from China, Korea, India, Singapore, Taiwan, and Japan. Now we are focusing the limited topics. For example, number one is necessary for non-clinical evaluation of human cell therapy product to enter clinical trial by using the virtual cell product. Number two is a risk-based approaches for starting materials and comparability for mesenchymal stromal cells. We summarizing our way of thinking in each Asian countries to make our deliverable publicly available to suggest our way to go. Today, Dr. Sakurai from PMDA will talk about update on regulatory aspect of gene therapy product in Japan. There are 9 approvable products in Japan at the moment. 6 of 9 are cell type products, but now gene therapy type products are emerging. So Dr. Sakurai will present current situation and update on gene therapy regulation, including the Cartagena Act. Dr. Yoji Sato from NIHS will talk about point to consider on the quality of raw materials for the manufacture of cell-based therapeutic product. Dr. Sato will focus on the current attempt to avoid divergence and unreasonable rigidity in the interpretation of and operation of quality and safety guideline document for cell-based therapeutic product. Mr. Tanaka is a member of firm and from Astra's firm. We'll talk about the regulation in Asia, United States, and Europe on starting cell for cellular therapy product and risk-based approach. Here I present the comparison of regulation in each country and region from case study approach and so on. First speaker is Dr. Sakurai. I will make a brief introduction of Dr. Sakurai. Dr. Sakurai is Deputy Review Director of both Office of Cellular and Tissue-Based Product and Office of Vaccine and Blood Product at PMDA. He is in charge of reviewer of gene therapeutic product and Cartagena Act. Act regulations. He received a Ph.D. in 2004 from Hokkaido University. He specialized in biology and worked as a scientist of University of Wisconsin Medicine, uh, National Institute, Research Institute of Police Science, and Tokyo Metropolitan Institute of Medical Science until he joined PMDA in 2013. He currently gets involved in revision of ICH-Q5A viral safety evaluation and biotechnology product as a deputy topic leader of MHLW PMDA Japan. Okay, please, so Dr. Sakurai.
Good morning or good evening, everyone. My name is Akira Sakurai from PMDA. I'm in charge of Director of Reviewers related to Gene Therapeutic Project and Cartagena Act. So I'd like to talk about update on regulatory aspect of gene therapeutic product in Japan today. This slide shows today's outline. First, I'd like to show the current situation of gene therapy in Japan, including current development of gene therapy in Japan, gene therapy regulation, and approved gene therapeutic product in Japan. And next, I'd like to talk about update on gene therapy regulation. This table indicates the Act on Regenerative Medicine or Gene Therapy in Japan. In Japan, regenerative medicine means cell-based therapeutic or a product for human use. Cells means such as muscle cells derived from iPS or ES cells, or mesenchymal cells, stem cells. And ex vivo gene therapies means something like CAR-T cells and gene-edited cells by CRISPR-Cas9 or TALEN. On the other hand, gene therapies mean both ex vivo and in vivo gene therapies. And interestingly, in vivo gene therapies include not only viral-based gene expression vectors, but also oncolytic viruses or plasmid DNA and messenger RNA. And I said before, I'd like to talk about gene therapies, means this red area. Oops. Gene therapies are actively developing in Japan. This slide shows the numbers of PMDA consultation and IND submission related to regenerative medicine and gene therapy in Japan. You can see almost half of consultations and INDs are gene therapies. Consultations of in vivo gene therapies are a little bit more than those of ex vivo ones. On the other hand, INDs of in vivo gene therapies are less than those of ex vivo ones. Oops. This table indicates the industry-sponsored clinical trials for gene therapeutic product in Japan. They are only a part of industry clinical trial related to gene therapies in Japan. I picked them up from the clinical trial database, NIPH Clinical Trials Research or Clinical Trials Gov. As I showed circle graph just before, clinical trials of ex vivo gene therapies, something like CAR-Ts, are more than those of in vivo ones. Okay, you can see that. Okay, next, I'd like to talk about gene therapy regulation in Japan. Next, I'd like to talk about the red area, ex vivo gene therapies and clinical research. Clinical research of ex vivo gene therapies should be conducted under the Act on the Safety of Regenerative Medicine. Under the Act of Safety of Regenerative Medicine, clinical research are classified into 3 classes: Class 1, Class 2, Class 3, by the potential risks. This slide indicates the decision tree of classification of researches of regenerative medicine. Just see the black rectangle which says cells to which genes was introduced. I think ex vivo gene therapies must say yes. So that means almost all ex vivo gene therapies are class 1. Class 1 means high-risk group. Such kind of research should be evaluated by the special committee, and MHLW should get the opinion from Health Science Council additionally. When the research plan is accepted by both special committee and council, the researcher are permitted to start. There are 16 permitted clinical trials, including 2 ex vivo gene therapies. I'd like to talk about clinical research of in vivo gene therapies. In vivo gene therapy research should be conducted under the Clinical Trial Act. But the act is literally for general clinical trials. I mean, the act is not specific for gene therapies. So we understand the specific act is necessary. In this case, now on discussion. Okay, next. Finally, about Japanese regulation, I'd like to talk about the Act on Pharmaceuticals and Medical Devices. All medical products for marketing, including gene therapeutic products, are regulated by the PMDA Act. And recently, there are a lot of innovative medicines with new technologies and strategy. Such new modality product may not be suitable for traditional development schemes. So, we regulatory agencies are establishing the early access schemes to the innovative medicine. EMA has Priority Medicine Initiative, known as PRIME, and FDA has a Regenerative Medicine Advanced Therapy designation. MHLW and PMDA established 2 ways for innovative medicine. One is Sakigake designation system, and the other is conditional and time-limited approval. This slide shows the strategy of Sakigake. Sakigake, or in Japanese pronunciation, Sakigake, literally means top runner, or something like that. So, Sakigake holders have the duty to develop the product in Japan with top priority. Japanese government will also help on every process of developing, including basic research, clinical trials, approval reviews, safety measures, insurance coverage, and so on. It's a big advantage. Okay, next. Next way is here. The development of medicine spends a lot of time to confirm the efficacy and safety on clinical trials. And the development of regenerative medicine product or some medicines for extremely rare disease needs more developing time. I'm afraid it takes forever. If the result of the exploratory clinical trials indicate predictably efficacy and confirmed safety, the product could be approved temporarily. That is conditional and time-limited approval. The temporary approval product should be confirmed the efficacy within maximum 7 years. If it fails to confirm the efficacy, the approval will be expired. Okay, next is approved gene therapeutic product in Japan. Okay. Oops. Okay, approved product is this area. It's Kemura, one of the most famous CAR T cells in the world. In Japan, it was approved in March 2019. The T cells collected from the patient and are introduced chimeric antigen receptor, known as CAR. The CAR expressed T cells are injected back to the patient body to attack cancer B cell. And this product information, it's normal approval and classified into human somatic cell process product and CAR T cells and orphan product. Okay, next product is here. It's Cartagena, the plasmid DNA encoded to human hepatocyte growth factor. It was approved in February 2019. This product is expected to regenerate blood vessels. And it's conditional and time-limited approval and classified into plasmid vector product. Okay, it's Zolgensma, known as the most expensive medicine in the world. In Japan, it was approved in March 2020. Zolgensma is a viral vector carries the human cervical motor neuron gene. I'm sorry, but the English version report of Zolgensma has not yet been made public. I hope it's coming soon. And this product information is here. It's normal approval, classified into viral vectored product, and based on adeno-associated virus serotype 9, orphan product, and Sakigaki project. Okay, next, I'd like to talk about update of gene therapy regulation. The guideline of gene therapy. Recently, we published one revised guideline and 2 points of consensus. The revised guideline is critical for gene therapeutic products, so I'd like to introduce it later. 1 point of consider indicates the quality and safety aspect of cell therapy products for cancer immunotherapy, including CAR T cells. The other point of consider indicated gene therapies using gene editing, something like CRISPR-Cas9 and TAREN, or a zinc finger, something like that. And this slide shows a revised guideline. The guideline name is Guideline on Ensuring Quality and Safety of Gene Therapy Products. And this revised guideline indicates basic technical methods necessary for ensuring the quality and safety of gene therapeutic products. The important point is the scope of the guideline is both in vivo and ex vivo gene therapeutic products. The slide shows the chapters of the revised guideline. Chapter 3 is quality, including expression, construct, virus, vector, and characterization. Chapter 4 is non-clinical studies, including biodistribution, general toxicity, assessment of gene integration risks, and potential appearance of replication-competent viruses. And chapter 5 is clinical trials. And chapter 6 is the risk of transmission of virus vectors to third parties. The concern about transmission risk is related to Cartagena Act. Okay, so next, I'd like to talk about Cartagena Act. Okay, the true name of Cartagena Act is the Act on the Conservation and Sustainable Use of Biological Diversity through Regulation on the Use of Living Modified Organisms. Oh, very long. The Act is based on the Cartagena Protocol on Biosafety to Convention on Biological Diversity. You can think this right word, okay? And that's the reason why the act is called Cartagena Act. And the scope is living modified organism, known as LMO. And there are some difference between Cartagena Protocol and Cartagena Act. And medical products are out of scope of the Cartagena Protocol, but in scope of Cartagena Act, unfortunately, it's a little bit complicated. This slide shows examples of LMOs. Genetically modified bacteria and animals are in the scope. Interestingly, viruses are not living in biology, but living in Cartagena Act. It's living carefully. On the other hand, genetically modified animal or human cells are not out of scope. It's not living. I don't know why. And this table indicates, oops, sorry. Oops. This table indicates the classification of regulation of LMO. Type 1 approval is for deliberate release, meaning the use of LMO without preventive measures against their dispersal into environment. Point for review for type 1 use are environmental risk assessment and risk assessment for third party. For example, gene expression virus vectors for human use, like Zolgensma. Type 2 confirmation is for containment use, meaning the use of LMO while taking preventive measures against their dispersal into environment. Point for review of type 2 use is the reasonable system according to risk for using of LMO. Type 2 use is for manufacturing process. On April 2019, new consultation menu start on the appropriateness of application dossier for regulation on type 1 or type 2 use. If you have some concerns about Cartagena Act, you can use our consultations. Okay, that's all for today. Thank you for your attention. Thank you very much.
Okay, thank you for Dr. Sakurai. Thank you for the good comprehensive presentation of now current regulatory aspect of gene therapy product in Japan. So, so even in Japan, even in Japanese, so Cartagena law is a very complicated. So,
yeah, that's right.
So we hope so. New consultation menu will so good help for sponsors companies.
Yeah.
Okay, so. Next speaker is Dr. Yoji Sato. Dr. Yoji Sato is the head of Division of Cell-Based Therapeutic Products, Japan National Institute of Health Science. As a graduate student at the University of Tokyo and as a postdoc at the University of Cincinnati, he conducted researches on cardiovascular pharmacology and succeeded in establishing a variety of transgenic mouse models to elucidate mechanism of cardiac excitation, contraction, coupling, and heart failure. Dr. Sato's current research area is in the field of regulatory science for the quality and safety of cell therapy product. He is currently leading the public-private partnership initiative in Japan, so-called the major project for validation of test method for tumorigenicity assessment of cell therapy product in collaboration with the Committee for Non-Clinical Safety Evaluation Pluripotent Stem Cell Derived Therapeutic Products Forum for Innovative Regenerative Medicine, so-called long name, FARB concept, and the Committee for the Cell Therapy Tracking, Circulation and Safety and Health and Environmental Science Institute, Healthy Cellular Therapy Tracks. He is also serving as the vice chair of the database committee, the Japanese Society of Regenerative Medicine, which provide a national regenerative medicine database, nationwide patient registry system for clinical research, And so post-marketing studies on cell therapy product, and the topic leader of ICHQ5A, as well as a member of the technical committee panel on science and technology, Health Science Council, and Japan Ministry of Health, Labor, and Welfare. Okay, Dr. Yoji Sato, please.
Thank you, Max, for the kind introduction. Hello, everyone. First of all, I'd like to thank the organizers to invite me to this wonderful meeting. So this slide shows you Japanese guideline documents for the quality and safety of cell therapy products. Japan's good tissue practices consist of general principles for the handling and use of cell or tissue-based products and the standards for biological raw materials. Dr. Tanaka, the next speaker of this session, will introduce the details of the standards for biological raw materials. The guidelines for manufacturing and quality control of cell therapy products and the standards for manufacturing facility form the good manufacturing practices for cell therapy products and gene therapy products. So we also have seven technical guideline documents separately written for each type of starting cell materials. The 7 guideline documents describe the basic technical elements to ensure the quality and safety of therapeutic products derived from processing of autologous and allogeneic human cells or human stem cells. The guidelines also clarify differences with respect to data requirements and evaluation between marketing authorization application and application of a clinical trial for an investigational new product. For the latter, points to consider are mentioned to make sure if there are any quality and safety concerns that might pose an obstacle to initiate a clinical trial. The 7 documents emphasize that when conducting or evaluating tests or individual product, it is necessary to take flexible approaches on a case-by-case basis in line with the concept of the guidelines and on the basis of type, characteristics, and intended clinical use of the product in question. And that the reflection of scientific progress and accumulation of experience in the relevant field is always encouraged, of course. The 7 documents also emphasize that in the use of such advanced therapeutic products for treating patients, especially in first-in-human with severe and life-threatening diseases or injuries, the risk versus risk balance between with and without the advanced treatment should be also taken into account rather than just discussing a known potential risk of a product. A decision made by a patient after extensive informed consent should be a crucial element. So before talking about biological raw materials, I'd like to introduce a draft document called MCP, which stands for Minimum Consensus Package for Ensuring the Quality and Safety of Cell Therapy Products. This document is a draft guidance for users of the 7 guideline document, which I showed you in the previous slide, and was written by the original drafting group of the guidelines. In the R&D and review process of an individual cellular and gene therapy product, in order to accurately and rationally ensure the quality and safety, appropriate test and data interpretation should be conducted based on the risk of the product, which is assessed according to the type, characteristics, and clinical application of the product. So excessive tests and data should not be required. However, to ensure the quality, safety, etc. of the individual patient, individual products, sorry. It is not easy for the developers themselves to select the necessary and efficient models and to evaluate the data among comprehensive models indicated in the current guidelines. So this issue has become a bottleneck for development. So for more reasonable, efficient, and effective product development, it is useful to share technical element and basic concepts, which we call minimum consensus package, which will be common basis for anticipated most human cell-based products among the stakeholders. So the MCP describes basic technical requirements, concepts, and principles that are common to most of cell therapy products and aims to prevent the divergence of guidelines interpretation and operation by sharing the minimum necessary recognition, such as principle, background, objective, etc. So the MCP is not a summary of the guideline documents, but rather their user guide. This slide shows you the table of contents of the MCP, which consists of 7 chapters and 6 addendums. Since time is limited today, let me introduce addendum 2, which describes the quality of biological raw materials for human cell-based products. The addendum 2 consists of introduction and 5 sections, namely biological raw materials not considered as hazard, cases where biological raw materials are considered as hazard, evaluation as hazard of biological raw materials other than intended components present in the final products, risk assessment by evaluating hazard anticipated for biological raw materials to be used during manufacturing, and countermeasures to be taken when the hazards anticipated for biological raw materials used during manufacture cannot be completely evaluated. In the introduction, it is stated that various biological raw materials may be used as culture media component or as reagents in the manufacturing process of human cell-based products. The most significant safety concern for using such material is whether they cause any undesirable health harm to the concerned patient. The evaluation of the safety for human cell-based products means not only the evaluation for human cell-based products themselves, but that the safety is also evaluated for the biological raw materials used in the manufacturing process. Therefore, basic concepts, appropriateness, and points of attention for how to deal with the issue of safety evaluation of biological raw materials are detailed in this addendum. In section 1, biological raw materials not considered as hazards are raised as shown in this slide. Because of the limitation of time, I cannot explain in details that section 1 is biological raw materials not considered as hazard. And there can be 2 principal cases where biological raw materials other than intended components are considered as hazards to human cell-derived products as final products. The one is cases where a significant amount of a certain biological raw material exists or remains in the final product, and such a material is likely to cause undesirable health harm to patients receiving such product. Another one is cases where antiviruses or prions may contaminate the biological raw materials, and such infectious materials contaminate the final products, likely causing undesirable health harm to patients receiving such product. For the first cases, Evaluation should be performed for any possibility to cause undesirable health harm to patients concerned, referring to test results using the scientific knowledge and or scientific method at present, based on the amount of such biological raw materials present in the final product. If any safety concern exists, the risk and benefits should be clarified to demonstrate appropriateness of the use. And this should also be clarified in the informed consent procedures for the patient concerned at the time of application of the product. However, the above-mentioned procedures should not be applied if the safety of the final product has been evaluated in whole. In the second cases, in cases where any viruses or prions may contaminate the biological raw materials, and such infectious materials contaminate the final product, likely causing undesirable health harm to patients receiving such product, it is desirable to rule out from biological raw materials actually used and possible contamination of viruses or abnormal prions likely to cause undesirable health harm to patients based on the general scientific knowledge and the rationale at present. Regarding burden of proof and proving method for the safety of biological raw materials, the developers or suppliers of human cell-based products has the principal responsibility of the burden of proof regarding the safety of biological raw materials based on the evaluation of hazard and the risk assessment anticipate for such materials. In this case, the developer or supplier should prove the safety using trustworthy documents prepared by the provider of such biological raw materials, well-known knowledge, conventional use experiences, or test data directly performed by said developer or supplier as needed. Sometimes, the absence of infectious materials may be proved at the upstream of processes, whereas at other times, tests using the final product may eliminate completely the safety concern of biological raw materials during the manufacturing process. And generally, preparation of all the biological raw materials used in the manufacturing process of human cell-based products by the product manufacturer is rare. And such materials are purchased as products from reagent manufacturers in most cases, There are no problem when the evaluation of the hazard is possible, resulting in successful risk assessment and the elimination of the safety concern based on the product specification. However, problems cannot be completely solved in many cases, as you know, as the countermeasures for such cases. You may ask the manufacturer of biological raw materials to furnish the information or any certificates conforming to the handling of such materials, or to request the supplier to perform the acceptance inspection. Or you may also change the manufacturing methods to those not using such biological raw materials, of course. If the measures indicated in the above 1 and 2 are impossible, while the use of such biological raw material is indispensable and the anticipated risk is considered comprehensively tolerable in light of benefit of treatment, you should consult with the regulatory authorities like PMDA and MHLW based on documents issued by the manufacturer, etc., of such materials or well-known and trustworthy information. So in summary, as mentioned in the next presentation by Dr. Tanaka, there are differences in the context, content, and the legal status of biological raw material standards, leading to difference in the flexibility of regulatory decision-making between the United States, EU, and Japan. Therefore, there is a need to share methods to risk analysis and risk management planning for CMCs and GMPs, and considering the target diseases, number of patients, patient prognosis, and quality of life. And currently, as I showed you in the presentation, an attempt is being made in Japan to avoid divergence and reasonable rigidity and interpretation and operation of quality and safety guideline documents for cell therapy products by creating a guidance document describing common basic technical requirements, concepts, and principles. So in the last slide, I would like to thank all the members of the MCP drafting group, co-chaired by Dr. Takao Hayakawa with myself. Thank you for your attention.
Thank you, Dr. Sato. So thank you for the good and comprehensive and informative presentation. Next speaker is Mr. Tanaka, Associate Director, CMC Regulatory Affairs, Astellas Pharma Inc. He has started his career at Chugai Pharmaceutical, a member of Roche Group after finishing a master course of Graduate School of Agricultural Life Science, University of Tokyo. After gaining the career in Genentech and a startup company for cellular therapy, he joined Astellas in 2015. And he has working in CMC regulatory for gene and cellular therapy field. He is a member of Pharm Regulatory Harmonization Committee and the working group lead of APAC RM. Okay, Mr. Tanaka, please continue. Tanaka-san. I can hear you.
Okay. Okay, so can you hear me? Okay.
Okay.
Yeah. So thank you for this opportunity to share the Pharm APAC RM activities. I'm Toshimitsu Tanaka, a lead of APAC RM Working Group 2 and belong to CMC Regulatory Affairs Astellas. So firstly, I would like to introduce APAC RM briefly. So APAC RM stands for Asia Partnership Conference of Regenerative Medicine. The mission is to expedite the launch of regenerative medicine and cell gene therapy for people in Asia. So our purpose is to promote harmonization and optimization of the regulations and to maximize the opportunities in Asian market. So industry group of regenerative medicine and pharmaceutical joined to APAC RM from these countries and region, the China, South Korea, India, Singapore, Taiwan, and Japan. The working group 2 of APAC RM is a group for quality discussion. So we have focused on 2 topics in last year and this year, the starting cells for cellular therapy product and comparability. As cellular therapy products are very broad, so we focused on the allogenic and differentiated the mesenchymal stromal cells, MSC. So I would like to introduce our discussion on the first point today. So this slide shows member of APAC RM Working Group 2, the members who are expert for CMC development on cell and gene therapy product and came together from the 5 countries and regions as in this table. So firstly, we reviewed the regulation of eligibility on starting cells in Asia, EU, and the US. So required virus or microorganism are tested for donor screening are checked. And the referred guidelines are put in the table as well. So standard for biological material in Japan, the mentioned by Dr. Sato at previous slide is included in this table as well. Red square item is considered for a case study, so that I will explain later. So the test on the HIV, HBV, and HCV are mandatory for all countries. So EB virus, the parvovirus B19, and West Nile virus are not so common requirement, but some countries require to test on these viruses. Negation of infectious diseases by the interview, informed consent for commercial use, and so appropriate site for correction are very common requirement among the world. So that window period of consideration in the red square is necessity to consider possibility that the virus infection that spreads through donor screening test. It may occur if donor is infected with virus only a few days prior to donor screening test. So Japan requested to perform the second virus test after passing window period of the first virus test. So Taiwan does not require the virus test twice. So however, a doctor should take consideration of the window period of the virus test when donor interview. The US requires a second virus test only for anonymous semen donors. For EU, so that if the samples from a living donor undergo serological testing and are also tested by NAT for HIV, HBV, and HCV, indicates the retesting after a time interval is not required. So by the regulation review, so it was revealed that each country or region has its own requirement, perhaps based on the history of the healthcare, the governance of remedy, and the situation of the infectious disorders. So however, the cellular therapy products are commonly developed in a different country or region from the place where starting cells are procured. So we did the case study in this table. So the cells are procured country A and are complied with regulation there. Then the cells are transported to country B and are developed in the country B. So country B requires the HTLV test for donor screening and window period consideration. So however, it assumed that due to the gap from the regulation of the country A, the HTLV test and the window period consideration were missed. This is the assumed case for this study. This slide shows an overall manufacturing flowchart of MSC product. So in this case study, donor screening and procurement of cells are performed in country A. Then transferred to country B and master cell bank is established there. The product is manufactured from the master cell bank and filled in a bag for development in country B. The 3 points for risk assessment are proposed in our case study. The 1, the donor location, and the 2, window period of the virus test, and 3, additional virus test on master cell bank. So this slide shows the geographical distribution of areas with high prevalence of the HTLV1 infection, which is reported by ECDC technical report. In Asia, country or region with high prevalence is Japan and Taiwan. And so those with low prevalence include China, India, and Korea. Though China shows low prevalence of the HTLV overall, but there are certain areas showing high prevalence. So therefore, if country A is low preference country, it is the low risk of the HTLV infection with procured cells. Regarding the window period of virus test, the EU directive describes that if the samples from a living donor undergo serology test and are also tested by NAT for HIV, HBV, and HCV, the retesting after a time interval is not required. The upper table shows comparison of the window period between the PCR test and also serologic test. As shown here, the window period of the PCR test is shorter than serologic test. So thus the risk of the false negative due to the window period of PCR is relatively lower than that of the serological test. And so this risk can be mitigated by the combination of the both tests. The lower table shows improvement of PCR test. Window period in 2017 is shorter than that in 1997. The 7 days shorter for HIV and the 17 days for HBV and 20 days for HCV. So therefore, the risk to miss the virus infection during the window period of the PCR test can be reduced by the recent improved the PCR method. And then we researched the virus tropism, virus receptor, and so propagation in MSC from literature. It was reported that cytomegalovirus and HTLV1 infect MSC and propagate in it. So therefore, additional test on master cell bank to detect these viruses is reasonable when missing the during donor screening test. For West Nile virus, it was reported that this virus that can infect very various cells. So however, we could not find out the investigation of infection with MSC. HIV and HBV and HCV do not infect MSC. So however, the test can detect that those viruses at least existing in culture media. So assuming that none of these viruses are inside of MSC that due to lack of the infection are related to MSC cells. In addition, HIV, HBV, and HCV are required to be tested at donor screening test in all guidelines. So therefore, the infection of the HIV, HBV, and HCV should sufficiently be controlled by donor screening test. As conclusion, the through case study, so we propose 3 points for risk assessment. And the first one is the donor location, and second is the window period of the virus test. And so the third one is the additional virus test on master cell bank. So the regulation is not harmonized, but the flexible operation of regulation, based on risk-based approach, with the close communication between health authorities and developers is very important to accelerate development of cellular therapy product in the world. Yes, thank you for your attention.
Thank you, Mr. Tanaka. Thank you for the good comprehensive presentation. So as all members knows, this topic, harmonization of these biological raw material regulation standard is very complicated. So we use the case study strategy. It's always people say it's always case by case. So it's very complicated. But we believe that this strategy, case study thinking is very important for the move next situation for the future. So, but maybe so sharing information and so exchange opinion is important. And so building a common view of prospect and challenge for or against regenerative medicine is very important. We think that's a first priority issue. So we believe this activity should maximize opportunity in the Asian market and so promote new products available to Asian people, including Japan. And in turn, that would lead the creation of the market comparable to North America and Europe.